The overall goal of this procedure is to precisely cut the neurons of C. elegans with a laser in vivo. This is accomplished by assembling the axotomy system and precisely focusing, aligning, and adjusting the power of the laser. When worms are immobilized on a microscope slide, their neurons can be cut with precise spatial and temporal control. The extent to which severed neurons regenerate is then measured by scoring for growth cones and/or by tracing neurite lengths.
The main advantage of this technique over existing methods, like the femtosecond laser, is that the MicroPoint pulsed laser is a cost-effective system that is easy to set up and maintain, and is readily adaptable for a wide variety of applications.
Though we typically apply this method to study regeneration of the GABA motor neurons, the system can also be used to ablate other cell types, such as skin, muscle, or specific neuronal synapses.
This method requires a MicroPoint laser system. The laser mounts to the epifluorescence port of a compound microscope and includes a single dichroic that matches the fluorophore in the cells to be cut, a pulse generator, and a foot pedal to operate the laser. At a minimum, the microscope should be outfitted with a 100 x 1.4 Na oil objective and a 4 x objective, have a motorized stage with joystick operation, and be mounted to an air table. For illumination, a light supply with light guide and a shutter mechanism is attached to the MicroPoint laser. The shutter mechanism is useful to reduce the illumination intensity independently of the laser. A camera, which is connected to a computer, requires a frame rate of at least 16 Hz and must operate well in reduced light. The computer requires imaging software, such as Nikon Elements, to view the